I'm Wendy Hartsock, science and peptide enthusiast. In this episode of Exploration Science, Jonathan Steckbeck, the CEO and founder of Peptilogics, discusses the Peptilogics approach to peptide drug discovery and intelligently navigating peptide chemical space. Why don't we start with maybe you just telling me about you? So, who you are and what you're doing.
Sure. So thanks for inviting me. So I'm Jonathan Steckbeck, the founder and CEO of Peptilogics. My personal background, so I have a mix of business and science, trained as a protein biochemist, PhD in biochemistry and biophysics, associated with, again, Wendy said we could go super nerdy here. So it was on peptide lipid interactions that had a lot to do and was very relevant to our lead drug program. So my kind of personal journey to get there was a little bit backwards. So I, did an undergraduate degree in microbiology. I've always been interested broadly in disease, but specifically infectious diseases. And then after a few years of working, I did an MBA, and that was really a function of having interned at a biotech in college, and where I learned there that while the scientists get to do all the cool work, they don't always make the decisions on what work gets to be done. And so I wanted to, bring some of that and learn a little bit about at least the business language. Worked for a few more years, and then was a little bit backwards. Then after a couple of years with an MBA, decided to do a PhD. And the impetus for that was I had been in Pittsburgh for a number of years, really understood the research ecosystem, and saw that there was a lot of work. The universities do a lot of good work here, but not a whole lot of it gets translated out. And so the idea behind it was to, be able to do deep technical diligence on a project that I could then hopefully take out and turn into a company.
Wow. Yeah. So how hard was it to go from working in an industry back into an academic setting?
It wasn't as bad as it might sound. I have, I kind of have learned to maneuver between the two different worlds, reasonably well over time. I think always having that mind, though, toward, some kind of product, it has been helpful, right? I think, as far as particularly, kind of going into the PhD studies, again, having a very clear idea of wanting to, not just have a single product, but have be able to create something that would maybe change the way we think about designing drugs in this particular instance, gave a different focus and I think a little bit of clarity to what can be a meandering process otherwise.
Yeah, absolutely. So then, were you positive you were going to start a company after your PhD, or were you sort of like playing around with different ideas?
So that was always the intent from the beginning, was to find something to then be able to take out. I think there's always a little bit of, it's not a, kind of, let's say broadly, entrepreneurship is I don't want to call it scary, but it's a daunting process, right? So there's always a little bit of, is this the right thing to do? But, at the end of at the end of everything, it had become very clear to me, based on what I wanted to be able to do, that starting a company was the best way to actually have a real impact and make ideally make a drug and then multiple drugs that would be able to help patients. And there's, the specific things that we work on, and I guess we'll come to this at some point, we're informed by, some things that had happened kind of personally. So.
Right. Well, yeah, maybe tell me about your company, Peptilogics.
Yeah. So Peptilogics, what we do is, and it's hopefully no surprise, we're a peptide-focused company. It's right there in the name. But the other thing that we do is also in the name is we really focus on a design-based approach. And so this stemmed from, as I alluded to, some of my work in my graduate studies, which was around trying to understand protein or peptide lipid interactions and how kind of structure function could change those. And, from a kind of a particular product or a future product standpoint, that was really evidenced by, or kind of really ensconced in this idea of what the molecule that became our lead program that we call PLG0206 now. And it was a peptide, as you know, short synthetic peptide that we designed to try to overcome some of the limitations of the molecules in its class. So broadly, it's an antimicrobial peptide. Going back, 40 years at this point, these molecules were discovered. They were going to be the next big class of kind of antibiotics. And the intervening 30 years had kind of told us, no, it's still drug discovery. It's still pretty hard. And what a lot of the challenges in that particular space were around, not that tox is ever wanted, right? But unwanted tox, right? And so we had been doing some studies on, structure function studies, trying to understand how we could take these peptides and modulate their activity, all the while keeping an eye on some of the programs that were more advanced, particular peptides that were being advanced, through different companies toward, clinical applications. And then just paying attention to the fact that, a lot of them fell out because of these tox problems. And so, after a while, we had a good understanding of that structure activity to modulate, again, kind of the antibacterial effects. And as we kept seeing this, idea of, inflammation being a problem with the broad class, taking kind of a deeper scientific understanding of what are the causes of inflammation. So inflammation is usually a receptor-driven process, right? And so there's some specificity involved. And so what we had learned from our structure activity studies was that the physical chemical properties of the molecules were important. And so what we decided to do was basically design a molecule to have the physical chemical properties that we wanted to be able to get the activity, but do it completely synthetically. So remove the sequence specificity of the broad class to try to address those particular tox challenges. So we did that, and that's kind of led to our lead program. And what we're doing now, well, maybe I'll just, before I move on from that, so that molecule is in a phase 1b proof of concept study now, and we can dig into the specific indications. But as the data, the only way to prove things in this kind of line of work is to actually do the studies, right? And so as those data were, continuing to be positive, they were showing us that the molecule, remained active, but didn't have any of the tox problems that were associated with the class. That gave us, through humans, right? So we did a human, first in human healthy volunteer study, intravenous delivery, set a very high bar for safety, and came through that really cleanly. So none of the challenges or none of the problems that were associated with this class were evidenced in 206. And so as we got those data, now what we're doing is, using a computational approach to take that same kind of understanding. Because if you think about what we did for 206, it was this idea of understanding how activity and then maybe tox related to the sequence and structure of the peptide itself. And so now we're using modern machine learning approaches to try to understand more broadly how different pharmaceutical properties, how more than just two pharmaceutical properties relate or can be related to sequence and structure to be able to design molecules, right, with the characteristics and the attributes that you want at the beginning of the process, rather than having to go through a number of iterative approaches, which have kind of led to the inefficiencies, let's say, of the discovery process.
And I do want to talk about some of the other areas, but going back to, and sorry, PLG 0206. Of course,
we can just call it 206. Yeah, that's what we do. It's a mouthful.
Nice. Yeah. I was looking at the sequence in a publication. So that looks like it's all L amino acids.
Correct.
And it's, maybe you could describe it rather than me describing it, since it's.
Yeah, sure. Yeah. So so 206, it's a 24 residue peptide, as when you said, all L amino acids, and there's no specific modifications to it. It's, sequenced. It's only 3 different amino acids, sequenced in a particular way to give us the characteristics that we wanted. So the activity characteristics, but by doing that, by using only 3 amino acids, we removed all the sequence specificity from it. And there's no specific, no special modifications. And so, part of what we wanted to do was, like I said, address the activity and the tox, The other big challenge, which again is probably no surprise to anybody listening to this, is the PK of peptides, especially intravenously delivered, has generally been thought to be challenging. Let's say short half-life, et cetera. And so what we also wanted to do was try to design some features into the molecule that might extend its half-life through non-traditional mechanisms. And I'm sure your next question will be, well, what is that? Well, what was that? And so
one more thing I just wanted to point out about the sequence is one face is all arginine and the other face is tryptophan and valine. So just so the listeners have an idea of what this molecule looks like.
Yeah. So it ends up being, so for this class of drugs, across this, regardless of what the specific structure of it is, and ours is an alpha helix, there is this amphipathic behavior. So all the charge is located on one side of the secondary or tertiary structure, and all the hydrophobic face is located on the other. Those tryptophans do end up being pretty important, though, just given where it tends to sit.
And so then, maybe you could now talk about, the specifics of the PK, because it is surprising, the half-life.
Yeah. So what we, again, kind of the general and some of the early conversations that I had, when I was trying to get Peptilogics started, talking to investors, et cetera, was that peptide PK was challenging, right? So we were going to develop this as an intravenously delivered drug for treating drug-resistant bacterial infections. And the expectation was that the PK would be low. We had animal data that suggested that it wasn't. And, you had to go through all the rigmarole of proving that out, right? And so doing PK studies in animals. But really what ends up happening is that the peptide has a reasonably high protein binding, but it's low affinity. And so what we ended up doing was, or what happens is that, the plasma proteins act essentially as a protective sink. So it keeps it circulating, right? It protects it from, kidney, filtration through the kidneys, but also protects it from proteases. The consequence of that at the end of the day is that, in our human study, we added, a 15-hour half-life. So, go from that kind of expected 2 minutes is what I think I heard the most often to 15 hours was something that we're pretty happy about.
Yeah, it's remarkable. And like you said, to do that through sequence design. So maybe going from there, you could talk a little bit about what the peptide's used for as well, because we know it's an antimicrobial peptide.
Yeah. And so, so we're developing 206 to treat periprosthetic joint infections. And now for us, again, one of the things we wanted to do early on was, we have 206 is a broad spectrum, so it kills both gram positive and gram negative. It's also a broader spectrum in the sense that it kills metabolically active and then inactive bacteria too, which is important. And I'll come on to that when we talk about the particular indication that we're treating, but it's truly a broad spectrum molecule. And so early on, we were trying to figure out what we would do. And the thing that made the most sense at the time was treat, drug-resistant bacterial infections. I think everybody understands that drug resistance is a big problem. The challenge became, and, over time, as we, again, kind of monitored of our space, our environment, it was clear that while antibiotic resistance is a big problem and it's something we are going to have to figure out how to address, the commercial prospects there aren't very good. It's a difficult place to have a commercially successful drug. And so we had some interesting data from one of our collaborators at the time, who was an orthopedic surgeon, and he was using 206 to treat these infections associated with prosthetic joints. And he's the most excitable guy you'll meet. He's just so ebullient. And he was so excited about the data that he was getting because, one of the challenges in periprosthetic joint infection is that surgeons, this is a problem we've had since we've been doing joint replacements, and it's the most devastating complication associated with them. And that's because we don't have an effective way to treat them, despite surgeons and ID docs doing everything they can think of to try to treat these infections. So whether it's, what we do now is try to cut it out, because over the past 30 or so years, intravenous antibiotics don't work. Dumping super high concentrations of antibiotics right on the prosthetic itself before they close up don't work. Alcohol, peroxide, you name it. None of them really have much of an effect. And what that ends up being, because of this, the way the bacteria grow, which is this biofilm-based infection. And why that matters is they're these complex communities, let's say, of bacteria, and they're composed of bacteria that are behaving, a couple of different populations of bacteria that are behaving differently. Some of them are actively growing and kind of maintaining the colony, if you will. And then some of them, about 5 to 10%, are really doing nothing, right? They're just sitting there inert. They're just kind of freeloading off of everybody else. Now, those are the challenging ones, because, typical antibiotic. So maybe just if I describe a biofilm a little bit. So it's this kind of, it's like a slimy community, right? So the bacteria secrete all these different, biomolecules. So there's, DNA, RNA, proteins, saccharides. There's all these different things composed in this, and they live in it. And so it provides a little bit of a structural barrier to the immune system and to some of those types of things, right? If you think about what are those, like Jello molds with fruit floating in them? So it's kind of like that, right? You've got little pieces of fruit in this Jello that protects it from kind of physical assault. Now, those freeloaders end up being the challenge for treatment with drugs, because most antibiotics inhibit some metabolic process. So either they inhibit cell wall growth, they inhibit, DNA replication, RNA replication, et cetera, et cetera. And so they work really well. They work really well on growing bacteria. It's why antibiotics have been and are so effective. On the freeloaders, they don't work because they're not growing. And so this is where 206, when I talked about that really broad spectrum, is important, because its mechanism of action is a little bit different. So antimicrobial peptides in general interact with the cellular membrane, so the bacterial membrane, and they act in some way to disrupt it, right? And we can come on to the specifics of what that looks like for 206 if it's important. But what that means in the context of these types of infections is that it doesn't matter what the growth state of the bacteria is. 206 will kill the actively growing in a biofilm, and they'll also inactivate and kill the freeloaders as well. And so it's really this comprehensive solution, which is ideal for these biofilm-based infections that occur on the joints.
And this doesn't require IV dosing to get to the joint. You can do direct. Go ahead, sorry.
Yeah, no, that's actually super important for. It's nice to that's not the case, but it's also important in this indication because that won't be how it's delivered, right? There are, the joint isn't well vascularized. So regardless of, what molecule you put in there, it's not going to you're not going to really get kind of the target attainment levels that you would need. It's also very difficult to convince a surgeon whose job it is to cut out the infection, because that's just what we do now, that they don't have to cut. And so really the benefit of 206 and why we started going down the road of treating periprosthetic joint infections was because the characteristics of the drug itself matched the treatment protocols. And so the two things that end up because of its mechanism that are true for 206 is that true broad spectrum activity. So it can be used empirically, because what we didn't talk about is that biofilm-based infections are incredibly hard to culture. And so most of the time, the surgeon has no idea what the infecting bug is. And so you have to have something that can treat it while they're doing the surgery. So it has to be, be able to be used empirically and be effective empirically. So cover everything you might see. The other thing, because it's a surgery, it has to be really fast. It has to work really quickly. And so again, because it's this mechanical type action, it changes the properties of the membrane. We've shown in vitro and in vivo, and we're testing it now in humans, that within 15 minutes, you can inactivate all of the bacteria associated with the biofilm. So the surgeon doesn't actually have to change his workflow, because what he's doing is, kind of opening the joint again surgically, and then cutting away the infected tissue and trying to just power wash off the prosthetic. And that doesn't work most of the time with what we have. And so the failure rates are like 60%. And so what we're doing is, instead of just giving him a saline wash, because like I said earlier, they've tried everything else and none of it works. So it's kind of counterindicated now. If they just use a 206 wash for 15 minutes, just kind of bathe the prosthetic in the 206 solution, all of our data so far suggest that you can inactivate, all of the bacteria, let's say, all the cultural bacteria within a 15-minute exposure. So it's important to match those two things.
That's great. And if anyone wants more information on 206, they can find it on your website if they want to get into the press releases or any of that. So now I'd love to kind of learn more about what you're using, the knowledge you've gained from that 206 program for some of your other programs.
Yeah. So 206, like I said, it ended up being the evidence and kind of the first lead, not just for our clinical programs, but how we think about arriving at drugs. And so what we're doing now is, and what we've been doing for the last few years, is building a computational system, a computational approach to really do the same kinds of things that we did with 206 in the lab and kind of in one person's head, to be able to make it a little bit more scalable. And so it's taking the best of modern machine learning, combining it with, in-house computational capabilities, and then all of the data that we can find to try to understand at a really deep level what kind of biological activity means. So maybe as a way to think about it is some of the things that are in the news most recently, like this Chat GPT that's everywhere. And so what that is, people have taken all of the text that's on the internet and used machine learning and deep learning techniques to try to understand both the structure and ability to create content with language. It's essentially the same thing that we're trying to do with peptides. We just have a smaller vocabulary, right? So we're working in the context of amino acids, and we work kind of across the spectrum there. So, native, unnatural amino acids as well. We also look to modifications. And so what, again, at the highest level, what we're trying to do is understand how kind of biological activity relates to the sequence and structure of a molecule to be prescriptive in its use. And so kind of in a drug sense, the highest way I can say it is like, when you've got a disease and you think about how you're going to treat that disease, you build a target product profile, right? This is the way we think, all the different attributes that we want the drug to have. We want to be able to get to the point where we can start with that, kind of input that into the system, right? The platform, in concert with the different disease characteristics, and have a good understanding of the relationship between all of those things to be able to then design a molecule for that disease that has these certain characteristics at the beginning, right? So from the very beginning. That's at a high level what we're trying to do.
And that, the platform is called Nautilus. So this is something that folks can learn about a little bit on the website.
We have a little bit of information on the website as well.
That's great. Could you talk more a little bit about the programs that you're exploring in your pipeline?
Yeah. So we have our two most, we've talked about 206. We have another molecule that we're working on called 301, which is another designed molecule focused on, it has a little bit different characteristics than 206. It's antibacterial and anti-inflammatory. And so we're looking at developing that for the treatment of infections associated with cystic fibrosis. Their inflammation is a big problem. So if we can specifically reduce the inflammation, we might get better outcomes there. And then now what we're also doing is using what we've built as far as the platform with Nautilus to expand our indications beyond infection, right? So looking toward some programs in rare genetic diseases. So we have a program against autosomal dominant polycystic kidney disease. And then we have a couple partner programs in oncology and inflammation as well.
Yeah. So maybe, I hope you don't mind, I want to kind of veer off a little bit, because I noticed that Peptilogics is located in Pittsburgh, Pennsylvania. Mm-hmm. And I saw that there's this, a Pennsylvania Life Science group that you're on the board of. Can you talk a little bit about just the biotech scene in Pittsburgh? And is it vibrant? Like, just kind of give me an idea.
Yeah. So it's interesting, right? And so maybe I'll just go back a little bit. So I'm not a native Pittsburgher. I grew up around Hershey, Pennsylvania. I use that one because everybody's heard of Hershey chocolate. Nobody's heard of the actual town. That's irrelevant.
What is it called?
So Lebanon, Pennsylvania. So I guess its claim to fame is it was at one point in time David Letterman's, fake home office, right? When he used to do the top 10 lists, he would don't know how. Anyway, moved to Pittsburgh and at the end of the 1990s. And, like I said earlier, there's a lot of great research that goes on here and traditionally has gone on here with the University of Pittsburgh and the University of Pittsburgh Medical Center, or UPMC. It's a really vibrant medical research ecosystem. Over the past, let's say 20 some years now, there's been a shift in the vibrancy of the biotech scene. So as more and more folks have kind of done a lot of the same things that tried to do some of the similar things that I have, which is take some of the research that's here, build it up, and turn it into a company. It's now becoming a much more vibrant ecosystem. Now, I'll be clear, it's not going to rival and doesn't rival Boston or South San Francisco and won't anytime soon. It's a little bit different beast. But the ecosystem here is the strongest I've seen it since I've been here. And I think it's on a good trajectory to continue to get stronger as more people take this same kind of journey.
Great. So now to go back to Peptilogics.
Sure.
I'm curious to know, with all these programs and what you're learning about peptides, are you staying within a certain length of peptide, or are you going to, is there no limit to where you go? Will you be doing small proteins? Yeah.
I think we'll be guided by the thing that's best for that particular disease. Now, one of the reasons, I guess we always have a bias, and I think everybody does, a bias toward smaller if you can, just for all the reasons that, it's a little bit easier to manufacture, et cetera, et cetera, et cetera. Now, one of the things, and one of the reasons we started this, started kind of building Nautilus, was because the chemical space is enormous, right? It's beyond astronomical. The numbers become ridiculous very quickly. And we don't have a good way, using the most advanced techniques that we have, we don't have a good way to really effectively explore that space. And, one of the things we believe is that, if you imagine kind of chemical space as a room, we've really only effectively explored maybe one tiny corner of it, because that's just what we've done. And we don't know what's in the rest of it. And, we believe that there are very similar, let's say, activity characteristics or kind of molecular characteristics in lots of parts of that room. We just don't know how to get to them. And so part of the idea behind Nautilus is, again, by understanding the principles associated with biological activities and then maybe kind of removing the human aspect from it and not being prescriptive about how you have to get there, we might be able to find things, way in the other far corner of the room that we never thought to look at or never had any idea of how to get there. And so, if that exists in kind of, if there are multiple of those new pockets that we haven't found, maybe some of them are smaller, right? So I know that's kind of a convoluted answer to the question, but it's, again, we always want to try to get to the best drug. And sometimes the best drug might be 50, sometimes the best drug might be 10.
Mm-hmm. Yeah, absolutely. Well, one thing that I noticed was, again, you already mentioned it, was how peptide forward the company is. So maybe you could tell me a little bit about, why peptides?
Yeah. Yeah. So I mean, for this audience, I think everybody gets it, but, for us, and then, the way I think about it, and when I started the company, it was really, if you take a step back and think about what we traditionally think of as drugs, right? So right now we'll set aside cell and gene therapies and some of the newer things like that. But you've got small molecules, and they exist on a spectrum, right? So you've got small molecules on one end, and you've got biologics on the other end, and somewhere in the middle fall peptides. And the exciting thing for us is that if you just kind of do a simple 2x2 matrix of kind of access versus specificity, small molecules have great access, right? They've been such successful drugs because they can get pretty much everywhere in the body. They do all the things that you want them to do. You can make them into pills, you can give them IV, whatever it is. But they don't have very great specificity, right? And so that leads to a lot of the side effects, et cetera. And, it's challenging to develop them as certain kinds of drugs as well, just because of size. On the other side, on the biologic side, they have kind of the opposite problem. They have the opposite characteristics, right? They've got incredible specificity. They do exactly what you ask them to do, but they don't get everywhere, right? And particularly, they can't get inside of cells, which is where a lot of what we think of as kind of undruggable targets live. Things that that's an attribute or that's a set of characteristics that leads to that, but that's their main problem, is they can't get into everywhere. And when you think about peptides, they can do all of those things, right? They have better access. There are peptides that get inside of cells. And they have, because they're biological polymers, they have very similar, if not the same specificity, or they can have the same kind of specificity that biologics have. Now, I think the challenge that the peptide industry has faced is that we don't know how to do some of those things systematically, right? Like, we don't know how to systematically make something super high specificity and get it inside cells if you want. That's part of the challenge that we're trying to address, because we believe of those three modalities, it's the only one where you can solve those problems. I don't think small molecules you're ever going to be able to solve. I'm kind of putting myself out here, right? There's, when it comes to biology, never say never. But, I think it's going to be very difficult to solve some of the challenges around specificity with small molecules. They're just, at some point, they stop becoming small molecules. And then on the other hand, evolution has made proteins and biologics to not be able to get inside things. Like, there's an outside and inside for a reason. We think that peptides are the only place that we're going to be able to, again, have the best chance of understanding how to specifically do both of those things at the same time. So kind of put us in the upper right quadrant versus one or the other.
Right. Yeah. Was there anything that I didn't ask that you wanted to talk about platform or programs or just Peptilogics in general?
That's a big question. No, I think maybe if I try to tie some things together, I think maybe through our journey, right? So we started with 206 as this thing that we designed to address some of these problems. It continues to advance. And now, like I said, we're in a phase 1B proof of concept, treating patients with prosthetic joint infections, and looking for a data readout here in a few months. And so that program itself continues to go really well. And then as we've now and continues to provide those proof points, right, for this idea of if you have a good understanding of what you want the molecule to do, or you want the peptide to do, maybe you can actually make it do that. And then following that with and building off of what we've learned there to broaden our approach. Because again, I think, we want to be able to do this not just for one type of disease, but for any type of disease. And as we advance some of these programs, whether they're our internal PKD program or some of our partner programs, we're starting to see that evidence itself, right? So the idea that you can understand biological principles and start to be prescriptive about how you encode them into a peptide, the early data there are good, right? And so the computational team has done a lot of foundational work on building, peptide specific models, because I think that's an important thing to think about too, is that, we're having to, we don't have the luxury that you have in small molecules of downloading drug bank and just, building some models and saying, okay, now I can do these things. We've had to be very thoughtful and very specific about how we approach it computationally to be able to understand the unique attributes of peptides. Now we've done some work to be able to pull from small molecules, because if you think about functional groups, those are the same, right? A methyl group is a methyl group. What matters is, can you describe the context in which it exists well enough to be able to pull from a small molecule and apply it to a peptide? And so we've done a lot of foundational work on how you do that. And that's actually really hard. That's actually really hard to get a computer to understand not just three-dimensional positioning, but three-dimensional positioning in an environment. And so a lot of the foundational work that our computational team has done is build the architectures to be able to do that. And I think we've been very fortunate to find an incredible team there, led by our chief technology officer, Nick Nystrom, who has a long history and is one of the world's best people in integrating machine learning and AI with purpose-built computing, because it's important to be able to do the right types of things at the right time. It's also happily cheaper than just trying to use Amazon for everything. But being able to do that has allowed us to do a lot of the things that we wanted to do, and will position us to be able to do it in the future. But the early data are good, and we've been pretty happy about that.
All right. I always like to ask big questions. Sorry, I can't help myself. So
no, it's good.
Another big one is what are some of the things that surprised you, or something that you learned that you couldn't have foreseen coming, again, from PhD and then starting your own company and getting to phase 1 and beyond?
Yeah, I think, the interesting thing, and I spend some time talking to PhD students, because, I think it's changing a lot more now, and I'm not that old, but the predominant, kind of moving towards industry, or particularly starting a company, wasn't something that was, particularly in the life sciences, a well-discussed career option. And so one of the things I think that I've learned is that it's kind of funny that PhD training is actually really good training for entrepreneurship, because you have they're not the same problems, but you have a lot of very similar challenges in that there's a lot of unknown. You are trying to work within that uncertainty to arrive at the best decision to be able to move things forward, which is, again, they're very parallel paths. One is a project, but you can look at a company kind of like a project. It's bigger than that, and it has more going on. And I don't mean to reduce it to such a simple thing, but a lot of the lessons that maybe the lessons learned, but also the characteristics I think that make a successful PhD student are important in starting and building a company. Like that curiosity to figure out, how do you address this problem, and the willingness to be able to do it in a new way. Try to take everything that you can and everything you can understand and synthesize it down into a way to address the problem. I think it's an underrated and not well discussed benefit of PhD programs. So I think that's the most surprising it took a long time to it took a long time to realize that, which is what I like to tell kids, tell students now.
Mm-hmm. Yeah. All right. And then last big question is, I always like to ask people, so what are you kind of most hopeful for, or hope will happen in the next 5 years? And this could pertain specifically to Peptilogics or to peptides in general. And you're not limited to one hope.
Yeah. Well, no, I appreciate that one. So obviously, I think, the main thing I hope that we can, that 206 continues to work, because I think it's one of those. Again, we didn't go a lot into the disease and the kind of the patient impact of the disease, but it's a really, maybe the easiest kind of way to contextualize it is it's a condition nobody's really ever heard of. I hadn't before we started doing it, but the 5-year mortality is worse than all of the cancers that you've ever heard of. It's worse than breast cancer. It's worse than prostate cancer. It's worse than all of these things that we fear, but nobody knows about it. And so, we're working to change that. But we're also, I think at the same time, hopefully we can give these patients back their life, right? I think the orthopedic surgeon that we're working with said it best. He's like, when a patient gets one of these things, it's a life-changing event, because like there's pre-infection and there's post-infection, and post-infection is not great because we just don't have a good way to treat this. So I think that's the one thing, I'm really looking forward to is, being able to, help real people live better lives. More broadly, what I'd like to in 5 years see is a lot of the kind of speed and improvements that a lot of these computational approaches, and there's this kind of idea behind, is it machine learning? Is it artificial intelligence? And what's the right terminology? And I don't think any of that matters. But what, we'll just broadly call it AI. I think if we can recognize even just some of the gains that have been made in kind of text and some of these generative capabilities, right? Because that's at its essence, that's what we're doing is, you think about ChatGPT or maybe things like DALL-E and some of the art generators, it's making new things based on what you tell it to do. And that's what we want to be able to do. So, I hope in 5 years that what we've been able to do is do well enough so that when we start a drug program, we have a much better idea of what the success is going to look like on the other side. And so what that means is that, we won't have to test as many, or we'll have a better understanding of, kind of what its human characteristics will be with fewer animal studies, whatever it is, right? But just fewer drugs or kind of fewer molecules to get to a drug, get there faster, get there cheaper, get there more efficiently. Because ultimately, I think what we have to be able to do, that because, the path that we're on for the price of drugs is unsustainable, right? If you kind of look at those curves over the last 40 years, you went from in kind of inflation adjusted dollars from a couple $100 billion to well over $2 billion, kind of all in cost of development. If it continues that way, we just can't support that, right? Our healthcare system can't support it. So if we can address and pull out a lot of that failure and increase the probability of success, maybe we can, while still allowing for the profit that you need, be able to bring down the cost of drugs to be able to get things to people a little bit more effectively.
Yeah. Awesome. So something I just have to add, because I love the technology. Obviously, I'm a peptide enthusiast. Is that as you're talking about price of drugs and making things more efficiently, I think that also lends itself towards the rare diseases, personalized medicines, all of these things that make drugs better drugs, right? That make molecules better drugs. Mm-hmm. And specifically act on this person's type of disease, even though it might be, the same type of cancer, but it's this person's body, to be able to just selectively target them. But because of all these other, platforms that are being implemented, like you said, you bring down the price. I mean, it really expands the space that the drug hunters can get into. So very exciting.
Yeah, absolutely. And I think, I think that's a great point. And I think the one of the real benefits of, again, kind of using these holistic AI-based approaches, is that now we can maybe understand, or the computers can be built to understand what makes you you, right? And why then, or how could we specifically make a drug not for a disease, but for your disease? And that, I didn't bring it up, but one of the things we also like about peptides is their modularity, right? The beauty of peptides is that in that case, we already know how to build that thing, right? If I come in and I have a certain mutation that the computer says I need this particular drug, and it's the only one that will work. Well, we could make that if it's a peptide, right? We know how to do it. It's a polymerization reaction. It's not that hard. We don't need to figure out the synthetic pathways for a small molecule, and we don't need to build a giant fermentation. We don't need to build a cell line and build a fermentation system and all those things. We can just build them on the fly.
Absolutely. Great. Well, if you don't have anything else, I would just want to thank you for your time and for such a fascinating story. I definitely, wish Peptilogics all the best and look forward to seeing the data coming out.
Yeah, thanks, Wendy. This has been really great. Thank you for having me.
Thank you for tuning in to this episode of Exploration Science. For additional details on the topic, please see the description box for links to articles and other resources. If you enjoyed the interview, please like, share, and subscribe. As always, we welcome your feedback and suggestions, which you can leave in the comments section. Thanks again for tuning in.